So today I just wanted to start with if anybody has any questions about chapters 16 or 17 about DNA structure, DNA replication, transcription, translation, mutations. Our quiz for 16, 17 is set for next Tuesday, so a little bit less than a week on the 24th. And I have not put up mastering for those chapters. I can root around and see what I can find. Because I did ask for 2 labs this week. The DNA extraction, which I put a due date on as Wednesday, and I know that's midweek, and you guys know already that that's no, it's not a super firm deadline. And for the DNA extraction, photo only. I don't need you to answer those questions because they're silly. They're not even really important. So photo only of you with your extracted DNA would be great. And then for Friday, to try to do over the weekend, the chromatography lab. And the main reason for that is because, for 2 reasons. We would have done that with photosynthesis and looked at the pigments in plants, but the video wasn't ready at that time. So that's one method or one way to study differences in pigments. The key to remember with chromatography is it separates molecules, particularly proteins, by charge and by size, depending on what solvent you use and what size paper or what size column you use. You can do chromatography in glass columns as well with little tiny microbeads in them of different sizes. The other reason that chromatography is relevant for this part of the semester is that if you watched the HHMI video, you might have picked up the guy named Erwin Chargaff, who helped Watson and Crick basically figure out that adenine paired with thymine and cytosine paired with guanine, that the percentages of A and the percentages of T were almost equal in most cells, and the percentage of C was almost equal to the percentage of G in most cells. That guy, how he determined that, Erwin Chargaff, was using paper chromatography. So it's relevant in a couple different times in a couple different places in the semester. So since we have a lot of lab this week and a lot of sort of hardcore memorizing process, I didn't feel like I needed to also assign you a bunch of mastering homework, but I can add some mastering in because I realize this is going to probably be a tough draw on your grade this particular quiz. So that's all I have to say. Let's ask if you guys have any questions that you either want to put in the chat or that you would want to, anything you want me to go over, because Monday is going to be probably too late, right? So if there's anything that you looked at, read, and I realize you guys just took your quiz last Thursday, right? So yeah, Thursday and Friday. So you should be about with me, right? So anything? Nothing? All right, then I will just briefly mention, because I can't help it, that for chapter 16, the outline is very detailed, probably more detailed than it should be. I would focus on, I think I did add a question as far as the history of the discovery or of the elucidation of the structure of DNA. All of the folks that I listed there, starting with Griffith and his pneumonia in mice, Avery McCarty McLeod, Hershey Chase, Chargaff, Franklin and Wilkins, and Watson and Crick, all of those people, you should, it's not so important the dates, but for those people, you should be able to, let's say, match them with their contribution. So that's that whole first page, basically. And then except for the structure of DNA. For the structure, obviously, you need to know those features of the structure of DNA, of the DNA molecule, including complementary base pairing, right? So which we often refer to as it being double-stranded, or you'll see people abbreviate it DS like that. So A pairing with T, C pairing with G, and how that base pairing occurs with the hydrogen bonds, 2 here and 3 between G's and C's, and they should be dotted because they are hydrogen bonds. Ooh, think back to chapter 3 or chapter 2. The complementary base pairing, that it is 2 strands, that it forms a double helix, and that double helix is driven by the nature of the strands being anti-parallel. And that is key because if you've looked it over, you realize that that 5' and 3', those designations show up quite a bit. And it's because all of the enzymes, both that are involved in replication, transcription, and actually even the messenger RNA getting placed correctly into the ribosome translation, they're all dependent on what is the molecule, the structure hanging off of the chain of DNA or RNA. Is it a phosphate, a 5' phosphate, or is it a 3' hydroxyl? So all right, those are the big features of the structure. Then for the replication, actually, I'm going to do a new board for that. For the replication, it is important, even though it's not discussed in a super amount of detail in the videos, it is important that you understand the definition of semi-conservative replication, as well as the process of replication. And for that replication process, if I were you, it is a visual process. So there are sort of 2 components to it, I feel like. There's a list of the enzymes that are involved in order for both the leading strand and the lagging strand, and you need to know their names and what they do. And then there is the visual part of how it actually works, right, of the process itself. So for the enzymes, I've listed them. They're in your book in a table listed, right? And there's also pictures in your book. The drawing that I did in the video and then included on your handout is pretty comprehensive, and as basically how I'm going to ask you the questions. So that's, I would watch my video for that. Probably the one from summer is probably better than the one from spring, but I don't know why I put them both up there. I think that must mean that I started the summer one, or I started the summer one without covering maybe something about semi-conservative replication. And I thought, oh, I should leave that up there because it's in the other video. I'm not sure. Wouldn't hurt you to watch it twice. I would seriously, for study purposes, for replication, draw it yourself. Get yourself, literally, I know it sounds very 3rd grade, but get yourself a blank piece of paper and do what I did, right? And draw the DNA molecule a little bit open, and then more of it unwound and unzipped, and then more of it unwound and unzipped, and go through it. Even if it's complicated and you don't get through it each time, every time you draw it, it will help you remember the steps. Oh, that didn't work out so well because I like the top stand to be here. But anyway, I would draw it and say it. And that's how I remember it. And I was just thinking this morning when I was laying in bed, I was like, oh, I'm not going to do the replication picture today. And I thought, how long will it be before I can't do it? Because I don't go over it as much, right? So even me, having done it for 20 years, right, or more, every time when I see this picture, I start and I remind myself at the very beginning, right? I say, okay, helicase is at the replication fork. It's unwinding and unzipping. What does that mean? It breaks the hydrogen bonds, right? Gyrase further upstream. And I know I'm not using the right colors, right? To help with the supercoiling. And then I start thinking about, okay, where does primase bind? What is the primer? RNA. And then DNA polymerase 3 jumps on and it, synthesizes, and then polymerase 1. And so, I mean, it's no different for me, is what I'm trying to tell you, right? And you do need to know the pattern and the order for both the leading strand and the lagging strand, right? So I'm going to put that here, leading strand and the lagging strand. Exclamation here, I'll put it in red, exclamation points. So you need to be able to do that. And then that's pretty much the end of chapter 16, because we are not covering that last section about how the DNA is folded up and the discussion about telomeres. Come to genetics, learn more about it. Chapter 17, transcription and translation. Looks like I didn't print that part of my own notes. That's pretty bad. Here it is. So transcription translation for chapter 17. Again, I think the notes are very extensive. There are gaps in them so that you can fill in. And again, I'd watch the videos and fill in. And then I would practice. You are going to need a copy of the chart for the translation, so that when you get to that part of the quiz, you are going to need to have with you a printed or from your textbook. Probably a printed would be the easiest. You're going to need a copy of the messenger RNA codon chart. 2 amino acids. Right, so you're going to need, and that's in your textbook. You'll find that on page 341-ish. It's figure 17.6, I think. As far as translation in chapter 17 and transcription and translation, not the people for the history. You don't have to know them. Okay, so you don't have to know about the famous experiment with the mold, right, with Beadle and Tatum. So that you don't have to know that stuff. And then that's it. Oh, and then mutations is in that chapter 2. So there's kind of 3 blocks for chapter 17. Transcription. Right. So the template strand of DNA being transcribed into messenger RNA. So DNA to RNA. And we focus on messenger RNA, but remember, it's all the different types of RNA, even the small nuclear types and all the regulatory types of RNA. And then translation, and again, that is similar. There, I have typed out those 5 steps. You have to memorize those. You have to know them in order. You have to know what happens in each step. I would draw it and say it at the same time, right? Just like for replication, and it's exactly what I do too, right? I say, okay, the messenger RNA binds to small subunit of the ribosome. First transfer RNA with complementary anticodon and appropriate amino acid, which is going to be methionine, binds to the message. Large subunit joins. I mean, that's what I do. I do it just like that. There's 3 sites, E, P, and A. And I just keep reciting it over and over. If you go through all 5 steps, and if you do that like twice, 2 or 3 times, you're going to remember them. So for 17, transcription, translation, and then the last section is about mutations. And those, again, are mostly definitions, and you will need the chart for that as well for the questions on the quiz about mutations. And I think that's the end of that chapter. We talk a little bit about how the ribosomes are arranged inside both prokaryotic and eukaryotic cells. And then they bring up sickle trait again, because it's such a dramatic mutation that has such a dramatic effect on the phenotype of the whole organism. And it's a single base change. And now you're in a place where you can understand that a lot better. We mentioned it for the first time way back in chapter 5. You probably don't even remember. We mentioned it that long ago. And now you can really fully understand it. All right. After going through that, anybody have any questions? That's only going to get worse with more coffee. No, nothing. Silence, crickets. Okay, I get it. It's that time of the semester as well, right? We're getting to the last push. We've got about 4 to 5 weeks left. it's Thanksgiving, it's Christmas, right? And we're all thinking about all those things and how can we do it in COVID times? And I understand that. But we have to finish the semester. This is the last hardest section. So if you can just focus until Tuesday of next week, really intensely, the rest of the semester lightens up a little bit after that, dare I say. So that's one thing I want to tell you. Oh, and I think I posted an announcement about giving, and I wanted to talk about that a little bit. So in November, at Camden County College, we call it Giving November, and we try to contribute to as many good causes as we can. As faculty, we take on various departments and individuals take on families for the holiday time, for gift giving. Usually the foundation runs a poinsettia sale that's open to faculty, staff, and students. And I did mention about the food pantry in our in that announcement. And we have a food pantry. It's called the Marketplace Food Pantry, and there's a branch of it in Camden as well as in Blackwood that is run by Jackie Tenuto, and it's funded totally by donations. And so we have some corporate sponsors, but primarily to the food bank, it's people like you and me just bringing in a can of soup and putting it on the shelf. And when we were in person, on the second floor of Halpern in the science building, several of us would offer, if students brought in canned goods or dried goods or a gift card from ShopRite or whatever, or sometimes personal items like shampoo, deodorant, etc. We would give them extra credit points on the exam, 1 or 2 points on the exam. And it got to be to the point where so many of us were doing it that the Marketplace had to actually hire a student worker to come over every Monday and pick up all the things that our students had donated. And I think I mentioned in the announcement that so far since spring, since we couldn't do that anymore, the foundation arranged for micro donations, what they call as donation as small as $1. So I did put up the pathway for you guys if you're interested. I'm offering 3 extra credit points on the coming exam because it's probably the one you're going to need them for. And then if a lot of people participate, then of course, Michael, and then if a lot of people participate, then I will match your donation. So there's 80 of us, 83 students in Bio 1. So I was thinking, well, gee, if everyone even just gives $1 or if half the people give $1, then I'll make a matching contribution. And because I'm going to donate anyway. And then my students can feel like they're making a difference and helping people. It's students helping students, right? If you need help, if you are food insecure, please, please, please go to the food pantry, contact the Marketplace. They can help you. All right. And it's not just ramen noodles. Like we make a big effort to actually provide full meals if possible. Usually not with the meat part because we don't have refrigeration yet, but we're working on it. So please, 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 if you need food, if you are food insecure, contact the Marketplace. If you can give back, it's a great way to help another student, and you never know, you might be helping somebody in our class, right? Okay, so that's my public service announcement, and that's all I know for today. So